Three invisible networks: the gut and the brain, the coral and its algae, the tree and its fungus. Each one is real. Each one has been documented in peer-reviewed journals, confirmed by independent research teams, and described as a breakthrough in how living systems communicate. Each one has also been simplified into a metaphor, sold as a product, and defended long after the evidence stopped supporting the version of the story that made it famous. This is not a coincidence. It is a pattern, and it has appeared in every episode this channel has covered for the past 2 months. The pattern works like this. A researcher finds something genuine. The finding is complex, conditional, and limited to a specific context. A metaphor is invented to make it accessible. The metaphor is better than the finding. It spreads faster, sticks longer, and reaches audiences that the original paper never could. Industries notice. Products appear. The metaphor becomes the product's justification. And then, years later, a review paper or a meta-analysis arrives and points out that the original finding never said what the metaphor promised. By then, the market does not care. The metaphor has its own momentum. Start with the gut. In 1984, in a hospital in Perth, Australia, Barry Marshall drank a broth containing Helicobacter pylori. He was a young gastroenterologist. He had been trying for 2 years to convince the medical establishment that stomach ulcers were caused by a bacterium, not by stress, spicy food, or excess acid. Nobody believed him. His supervisor thought he was wasting his career. Pharmaceutical companies were earning billions from acid-reducing drugs that treated symptoms without addressing the cause. Marshall drank the bacteria. Within 5 days, he developed gastritis. He biopsied his own stomach lining, confirmed the infection, treated it with antibiotics, and recovered. The paper he published changed gastroenterology. He won the Nobel Prize in 2005. Marshall proved something specific, that a bacterium living in the stomach could cause disease. He also proved something broader, that the gut is not a passive tube. It is an environment full of microorganisms that interact with the rest of the body in ways the medical field had underestimated. That insight opened a door. Behind it was the gut-brain axis, the idea that bacteria in the gut can influence the brain through the vagus nerve, immune signaling, and the production of neurotransmitters like serotonin. Roughly 90% of the body's serotonin is produced in the gut. The bidirectional communication between gut and brain is confirmed by multiple research groups across 2 decades. It is real. What is not settled, and what matters enormously for what came next, is how much that communication can be manipulated with over-the-counter products. In 2011, a team led by John Cryan at University College Cork published a study showing that a probiotic bacterium called Lactobacillus rhamnosus reduced anxiety-like behavior in mice. The mice that received the bacterium were calmer, less stressed, and showed measurable changes in brain chemistry. The paper was widely cited. It was described in Popular Science as evidence that gut bacteria could treat depression and anxiety. The phrase, "your gut is your second brain," appeared in headlines, book titles, podcast episodes, and marketing copy. Probiotic companies referenced the gut-brain axis on packaging. The global probiotic market, already growing before the mouse study, accelerated sharply. By 2025, estimates placed it between $77 and $114 billion, depending on the source. Supplements promising to improve mood, reduce stress, sharpen cognition, and support mental clarity line the shelves of pharmacies and health food stores in dozens of countries. The packaging cited the gut-brain axis. The fine print, when it existed, cited mouse studies. The human trial came in 2018. The same bacterium, the same research lineage, Lactobacillus rhamnosus JB1, was tested in healthy adult males over 8 weeks in a randomized, placebo-controlled trial. The result? No significant effect on stress, mood, cognition, inflammation, or hypothalamic pituitary adrenal axis response compared to placebo. The researchers themselves noted that the findings in mice did not translate to humans. A pilot trial, another probiotic strain by the same group, also failed to replicate its animal results and was criticized for methodological fragility. The studies received a fraction of the media coverage that the original mouse result had generated. They did not slow the market. By the time the correction arrived, the shelves were already stocked. This is where the pattern becomes visible. The gut-brain axis exists. The specific claims made on probiotic labels largely do not. The gap between the two is filled by a metaphor. Your gut is your second brain. It is a compelling image. It implies that fixing the gut fixes the mind. The science says something more cautious. The gut and the brain exchange signals through pathways that are not yet fully mapped, and manipulating those pathways with over-the-counter supplements has not been reliably demonstrated in humans for most of the conditions advertised. The metaphor sells. The caveat does not. Move to the reef. Coral is not a single organism. It is a consortium. The coral animal, photosynthetic algae called zooxanthellae, bacteria, fungi, viruses, and other microbes. Together they form what biologists call a holobiont, a term coined by Lynn Margulis in 1990 and adopted by coral researchers in 2002. The symbiosis is real and foundational. Corals cannot survive long-term without their algal partners, which provide up to 90% of their energy through photosynthesis. The coral provides shelter, carbon dioxide, and nutrients. The algae provide sugars. It is one of the most productive partnerships in biology, and it built the largest living structures on Earth. When water temperatures rise by as little as 1 to 2 degrees above the summer maximum, the photosynthetic machinery of the algae begins to malfunction. The coral expels them. The tissue turns white. This is bleaching, and it has killed large portions of reef systems worldwide in events that now occur roughly every 6 years instead of every 25. The metaphor that grew around this biology is the superorganism. The idea that a coral reef functions as a single integrated system, resilient and self-regulating, provided external stressors are removed. Conservation campaigns adopted this frame. Media coverage declared the Great Barrier Reef dead, then alive, then dying again, as if an ecosystem the size of Italy could be reduced to a binary. An obituary for the reef, published in Outside magazine in 2016, went viral. It was satire, but it was shared as fact. The actual reef is a mosaic of over 2,900 individual reefs stretching 2,300 kilometers. Some sections bleach and recover within a decade. Others bleach and are replaced by algal turf that prevents coral recolonization. Recovery depends on local water chemistry, species composition, the interval between bleaching events, and whether sediment and nutrient runoff from agriculture compounds the thermal stress. The holobiont concept is scientifically productive. The superorganism metaphor is not. It implies a unity and a resilience that obscures the fragmentation the actual data describes. The contested part is not whether corals have microbial partners. They do. The contested part is whether those partners form a coherent evolutionary unit, a hologenome that adapts and evolves as a single entity. A team of evolutionary biologists argued in MBio in 2016 that this framework was based on overly restrictive assumptions, that a host plus its microbiome is more accurately described as an ecological community with a range of interactions from parasitic to mutualistic, not a superorganism with unified interests. The debate is ongoing. Meanwhile, conservation policy informed by the superorganism model sometimes treats reefs as systems that will bounce back once external stressors are reduced, underestimating the threshold effects and irreversible regime shifts that determine whether recovery is possible at all. When a coral reef crosses a tipping point, removing the stressor does not reverse the damage. The metaphor does not account for tipping points. The reef does. Now the forest. This channel spent 2 episodes on the mycorrhizal network, the science behind it, and the political war it fueled. The summary is brief. Suzanne Simard showed in 1997 that carbon moves between trees through underground fungal connections. The finding was real. The phrase coined by nature editors to describe it, the wood wide web, was a metaphor. Over 25 years, that metaphor grew. Trees became cooperative. Forests became communities. Mother trees nurtured their offspring. A best-selling book in 2021 turned the idea into a cultural phenomenon. Film rights were sold. The narrative was powerful, personal, and emotionally resonant. In 2023, Justine Karst and colleagues reviewed 1,676 citations of mycorrhizal network research. They found that fewer than half the claims made in papers published in 2022 could be considered accurate representations of the original data. The distortions moved in one direction, toward a more connected, more cooperative, more human version of the forest. The metaphor had become the evidence. Karst herself has said that mycorrhizal networks probably do play a role in some tree-to-tree interactions, and that better experiments could clarify the question. The problem was not the finding. The problem was the story. 3 networks, 3 metaphors. Your gut is your second brain. The reef is a superorganism. The forest is a family. Each metaphor contains a truth. Gut bacteria do affect the brain. Corals do depend on microbial partners. Trees do share a root environment with fungi. Each metaphor also contains an inflation. The gut-brain axis does not mean that a probiotic capsule treats depression. Coral symbiosis does not mean that reefs are self-healing systems. Carbon transfer between trees does not mean that forests are intentional communities. The cost is not abstract. When the gut-brain metaphor becomes a marketing tool, $80 billion flows into a market where most products have no demonstrated effect for the conditions they imply they treat. When the reef superorganism metaphor becomes conservation policy, recovery timelines are underestimated and threshold effects are ignored. When the forest family metaphor becomes a political weapon, logging companies justify selective harvesting by keeping mother trees, and conservation groups demand total bans based on a level of interconnection that the data does not confirm at that scale. There is a rhythm to how this happens. A researcher publishes a careful, limited finding. A science journalist or an editor translates it into a narrative with broader implications. A metaphor is invented. The metaphor is better than the finding. It is simpler, more emotional, easier to remember, and harder to fact-check. It reaches an audience 1,000 times larger than the original paper. Industries, advocacy groups, and policymakers adopt the metaphor because it is useful. It justifies products, policies, and positions that already existed but lacked scientific cover. The metaphor becomes infrastructure, labels, laws, donation campaigns, TED Talks, bestsellers, documentary series. And then, quietly, sometimes a decade later, a review paper appears. It says, "We checked the citations, and the original finding was more limited than the story that grew from it." The review gets a fraction of the attention. The infrastructure remains. The question this leaves is not whether metaphors are good or bad for science. They are both. The gut-brain axis metaphor brought funding and public interest to a field that had been neglected for decades. The Wood Wide Web made millions of people care about forest ecology for the first time in their lives. The holobiont concept gave coral researchers a productive framework for studying symbiosis that generated real insights into reef biology. Metaphors open doors. The problem is that they also furnish the room before anyone checks whether the walls are load-bearing. Once the furniture is in, removing it feels like destruction, even when it was never supposed to be permanent. This is the pattern that has run through the past 2 months of this channel. A bacterium that disproves a consensus. A reef that dies in public and recovers in silence. A forest whose connections are real but whose story outran the data. A plastic-eating microbe whose discovery became an alibi for the industry that created the problem. A wolf reintroduction that produced a myth worth $43 million. In every case, the finding was genuine. In every case, the version that spread was not quite the finding. And in every case, the correction arrived after the metaphor had already built something on top of itself. Whether that pattern is a flaw in science communication or an unavoidable feature of how humans process complexity is not a question with a clean answer. Science generates findings. Culture generates stories. The two processes run at different speeds, serve different purposes, and answer different needs. By the time they collide, the story usually wins. The findings survive in journals. The stories survive everywhere else. Next time, a doctor in Vienna discovered that washing hands between the morgue and the delivery room reduced maternal deaths from 18% to 1%. He was right. He was also, by every account, impossible to work with. The discovery did not save him. Whether it could have is still debated.